Hello dear students, in this video we're going to talk about our lesson, the transcription and translation, and we'll talk about some types of mutations. Let's start first about the chromosomes. The chromosomes, they are at metaphase. They are made up of two chromatids. These two chromatids, they form the chromosomes at the metaphase. The substance that make up the chromosome, as we said before, is called the chromatin. So the chromatin is composed of nucleotides. These nucleotides, they are A, T, C, and G. A and T, they are complementary. C and G, they are complementary. So here we have the complementary nucleotides A and T, and we have the complementary nucleotides C and G. So here, these nucleotides, they will form most of our work here. So A, whenever we have, let's say we have the sequence here, A, T, C, G, A, G, C, T, for example. So this is the first strand. Every one of these is coded in nucleotide. So A is coded in nucleotide, T is coded in nucleotide, C is coded in nucleotide. Every three of these, they are called codon, or they have another name, they can be called triplets. So codon or triplet. So A, the complementary of A, the complementary of A will be, for example, let's have the blue color. So the complementary of A, it's T. The complementary of T, it's A. The complementary of C, it's G. The complementary of G, it's C. A, T, G, C, C, G, T, A. Also, the chromatin is composed of proteins, composed of histones and non-histones. DNA, by the process of transcription. So we can move from DNA into the mRNA. Here in the nucleotides, we have something special. There is no more T. So T is going to be substituted by U in the mRNA. And by the process of translation, we have the polypeptide, which is chain of amino acid. Let's start with transcription. Transcription is the process by which mRNA is produced based on the given sequence of DNA. Where does this process occur? We said it occurs inside the nucleus. So what happened to the produced mRNA? They undergo mutation, and they are transported from the nucleus toward the cytoplasm. DNA can give mRNA under the action of two enzymes, helicase and RNA polymerase.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
So consider the sequence of the DNA molecule shown here. Here we have the sequence of the DNA molecule. Here we have the sequence which is called the non-template. First of all, you have to know that the non-template is called the non-transcribed, and it's also called the coding. So they gave you this sequence of the exercise. They said that we have the sequence of the DNA. They told you that this sequence, it's the non-template sequence. So the non-template sequence, they said write down the sequence of the mRNA molecule. Here we have two choices. Either we use this sequence, and this sequence is the same, the non-template, the non-transcribed, and the coding, it's the same as the mRNA. Instead of T is replaced by U. Or we have to get the complementary strand, which is called the template, also which is called the transcribed, also it's called the non-coding. So if you want to go to complementary, G, the complementary is C, G, T. T is the complementary is A, G is C, G. T it's, sure we have to put dash, A, T, C, T it's A, and C it's G. T it's A, G it's C, C it's G. C it's G, A it's T, T it's A, T it's A, and G it's C, and C it's G. If you want to calculate, or sorry, you want to find the sequence of the mRNA molecule, the messenger RNA, you have two choices. Either you use the non-template, or you use the template. If you use the non-template, which is the non-transcribed, the coding, the same sequence, but I have to replace the T by U. The same sequence, but I have to replace the T by U. Or if I use the template strand, which is the transcribed, the non-coding, I have to use the complementary, and instead of T, it's U. Here is the sequence. First, the non-template. The non-template strand is called the non-transcribed, and it's the coding, while the template is called the transcribed one, and it's called the non-coding. In order to write the template, you have to make the complementary strand to this. Okay, so the complementary for G, C, C, G, A, T, and so this is the non-template, and here I got the template sequence. mRNA sequence. If you have a look here, like GCA, GCA, TGC, UGC. So T it's replaced by U. ATC it's AUC. So T also is replaced by U. TGC it's either UGC, the same, but T is replaced by U. CAT, CAU, U and T. TGC it becomes UGC. So T is replaced by U. Here we can deduce the non-template, the non-transcribed, it's the coding, same as mRNA, instead of T is replaced by U. If I want to use the template, I have to make it complementary. So C complementary is G, G is complementary is C, T is complementary is A, while A the complementary is T, but here I put U. Pay attention to this. So mRNA strand is complementary to the template, while it's identical to the non-template where T is replaced by U. So this is an important helicase, I said, and wins the DNA, as you see in the video, coding for the given protein. RNA polymerase synthesizes the complementary strand. Second step of protein synthesis is called the translation, where RNA is going to code for proteins. These proteins, either they remain in the cell or they are exported out of the cell. Given the sequence is the coding. So it's non-transcribed, non-template coding. They wanted this transcribed, so I have to complementary. And if they want the mRNA coding is the same, instead of T is replaced by U. mRNA is the sequence, as I said, same as the coding, instead of T is replaced by U. How can I get the sequence of mRNA? It's not a secret. It's from the genetic code table. So here, for example, let's start with the letter U. You have to study only 3 of these: UAA, UAG, and UGA. These 3, they are the stop codons. And the others, you can use them. For example, first letter is A, second letter is C, and the third letter is C. So ACC, very unique, THR. Okay? For example, first letter is G, and the second letter is U, GUU, let's say. It's valine. Okay? So you have to use this genetic code table, and you have to study only the stop codons. Type of. In this video, we're going to talk about type of mutations in general. Next video, we're going to move deeply into details about the types of mutations. First, we have insertion, deletion, and we have substitution. So let's start with insertion, let's say. If you want to say insertion, okay? Let's say we have insertion. Here we have the sequence of the DNA template. So here I have to insert. So insertion of a specific DNA sequence. Let's say I want to insert here in this region, I want to insert A. So what the sequence will begin? It will be A, G. Then I inserted A, C, T, C, G, A, T, T, and we have C. So here we have 3, and the 3, and the 3. We're still 1 alone. This is called insertion. Does it affect the synthesis of proteins and others? We will talk about this in the second video next week. Second type of mutation is called deletion. For example, let's say I deleted the second nucleotide of the third codon. First, this is the first codon. This is the second codon. This is the third codon, and this is the fourth codon. We said codon is composed of 3. So the first codon, second nucleotide of the first codon, this is the first codon. Second nucleotide, 1, 2. This is the one deleted. So it becomes A. A is deleted, so we have to shift it here. So G, C, T, C, G, A, T, and then we have T, C. So we have 1 deleted nucleotide. What will happen? We'll talk about this next. The third type, which is the last type for this video, substitution. For example, I substituted the second nucleotide of the third codon. So first, second, third, fourth. This is the third. First codon, second codon, third codon, and this is the fourth codon. Second nucleotide of the third codon is substituted by G. So instead of A, I have to put G. So the sequence be A, G, C, T, C. G, instead of A, it's G, A, T, T, C. Substitution, they are derived into 3 types. We're going to talk about them next week. Okay? Thank you. Bye-bye.